Hi guys, this is Mrs. Meek, and today I am going to tell you all about squishy. Alright, not this squishy, which we know as a jellyfish. Alright, this is Dory squishy. Today we're going to talk about my squishy. Do you remember my squishy? Alright, when we talked about lipids, this is a phospholipid. And it is one of my favorite biological molecules because it makes up your plasma membrane. All right, so today we're going to talk all about Squishy and some of his friends. All right, so the whole function of the plasma membrane or the cell membrane, different textbooks use different names, but they're referring to the same thing. All right, so the plasma membrane helps to maintain a cell's homeostasis. It's a very thin, flexible boundary that allows nutrients into the cell, and it allows wastes and other products to leave the cell. All right, but it helps to keep and maintain that internal environment that the cell needs in order to carry out its job. All right, this boundary is what we call selectively permeable. All right, when you think about the term selective, all right, if you are a person that is selective when you choose friends, that means you're not just friends with everybody. All right, you have certain criteria or characteristics that you're looking for. All right, some people are selective eaters, which means they only eat certain kinds of foods. So selective permeability refers to the idea that the plasma membrane only allows some substances to pass through while it keeps others out. All right, when we look at the structure of the plasma membrane, all right, it is made up of lipids. All right, there is a layer, two layers of these phospholipids that are arranged tail to tail. We're going to take a look at that arrangement in a minute, but let's just review what makes up a phospholipid. We have a polar head, and then we have two nonpolar tails, which I like to call fatty acid tails. All right, so if you remember when we talked about covalent bonds, all right, this polar head carries a charge. These nonpolar fatty acid tails are composed of molecules that are not charged. All right, so the polar head is attracted to water, which is also polar. And the two fatty acid tails, which are nonpolar, are repelled by water. All right, so this allows for these phospholipids to arrange themselves in this bilayer, all right, because the environment outside the cell is mostly water and the environment inside the cell is mostly water. These molecules line up in a double layer where the polar hydrophilic heads are facing towards the water and the hydrophobic nonpolar fatty acid tails face each other. All right, so these phospholipids actually create a molecular sandwich that separates the environment outside the cell from the environment inside the cell. All right, so these phospholipids in large numbers will automatically make this arrangement when they are in a predominantly water or aqueous environment. All right, so in this plasma membrane, there are a lot of different components. It's not just these fatty acids. All right, there's other things that are also in this membrane that allow the cell to carry out a lot of its jobs. There's proteins, there's cholesterol molecules. You can see that right here. There's a cholesterol chain that's attached to this protein molecule. All right, there are carbohydrates, all right, that move in and among this phospholipid bilayer. All right, a part of these components include receptors. All right, these receptors allow the cell to communicate and transmit signals to other cells nearby. So when your body needs to make clotting proteins, all right, the one cell may get a signal and then it sends it to another cell. This is how cells are able to communicate. All right, we also have transport proteins. Transport proteins will span the entire phospholipid bilayer and they create tunnels through which substances can enter and leave the cell. We're going to talk more about that in the next segment. All right, but just understand that embedded in this phospholipid bilayer are a lot of other proteins and carbohydrates, other macromolecules that contribute to the ability of the cell to do its job. All right, many scientists refer to this lipid bilayer as a fluid mosaic model because it's not rigid, all right, it's able to move. So the structure of the plasma membrane resembles a C on which these other proteins and carbohydrates are able to float and move around. All right, so this is a natural tendency for these phospholipids. It's a natural tendency for them to pair up and make this sort of molecular sandwich. All right, but it's very flexible and it's able to move and it's able to carry out all the functions that it can carry out in this very simplistic design. All right, I have 2 video clips that I'm going to share with you that talk more about the wonderful world of plasma membranes and the phospholipids that make them up. I'll see you tomorrow in class. Have a great evening.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
The plasma membrane is made up of several organic molecules. I will concentrate on 2 of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycele would form. A mycele is a sphere with the hydrophilic heads on the surface, the tails inside away from the water. Our bodies are made up of approximately 60% water and trillions of cells. The water can be found inside the cells, outside the cells, and in the bloodstream. The location of water and the phospholipid, hydrophilic, and hydrophobic ends enable the membrane to form a bilayer of lipids. The currently accepted model of the cell membrane was developed by Singer and Nicholson in 1972 at the University of California, San Diego. Pictured here, the plasma membrane looks like a sea of lipid with icebergs floating in it. The membrane is very flexible and has a fluid motion. This design of the plasma membrane is called the fluid mosaic model. Quite often, these proteins have sugar molecules attached to them, forming glycoproteins. These molecules extend from the membrane surface and act as an identification marker. The proteins are used by the immune system to identify a cell as being self, or foreign called non-self. Each individual has their own biochemical makeup. In other words, each human being has their own set of cellular proteins. But if a cell comes into the body that has a different set of proteins, whether it is a bacteria or an organ transplant, the immune system may identify it as self or as non-self and attack. In order for the cell to survive, many different materials must be able to cross the plasma membrane. When water passes through a cell's membrane, the type of diffusion is called osmosis. When solute particles pass through a cell's membrane, the type of diffusion is called dialysis. Before looking looking at this movement through the membrane, it is important to understand the movement of molecules in general. Atoms and molecules are in constant motion. This random motion will disrupt the cluster rather than maintain it. This motion is referred to as Brownian movement. This motion is always present in some degree, in gases, in liquids, and even in solids. For instance, if a bottle of perfume is left open in a room, eventually, no matter where you are in that room, you are able to smell the perfume. The molecules of air hit the liquid, causing the liquid molecules to move into the air. At the time the bottle is first opened, there is a high concentration of perfume molecules in the bottle, a very low concentration of perfume in the air. As the perfume molecules are bombarded by the air, the concentration of perfume is dispersed into the air. In other words, the perfume is moving down a concentration gradient, high to low. This movement is called diffusion. It uses Brownian movement. Now let's take a look at another example of diffusion. In this aquarium of water, we drop some blue dye. As the water molecules move, they bump into the dye molecules. The dye molecules, at first in a high concentration, start moving and bumping into each other and the water molecules, moving into the area of lower concentration. As this movement continues, the dye spreads out and equilibrium is attained. Equilibrium is equal concentrations of the dye and water in every area of the aquarium. Remember, Brownian movement continues even after equilibrium is reached. Although extremely thin, the plasma membrane separates the contents of the cell from the environment. The membrane has several functions. This video concentrated on the movement of materials through the plasma membrane. Remember that this membrane is selectively permeable and can prevent materials from passing through or keeping materials in, and it will permit, even facilitate, the entrance and exit of materials. Materials may move through membranes passively or actively. In this video segment, passive transport, which uses Brownian movement, was introduced. Passive transport includes diffusion, defined as the movement of materials from a region of high concentration to low concentration. Two specific forms of diffusion are osmosis and dialysis. These processes are essential to the survival of the cell. Nutrients must enter, wastes must exit. The structure of the membrane allows for the processes to take place, which enables the cell to live.